Hello and welcome to my channel Omni Bio Entrance, and today we will be discussing DBT JRF entrance exam question paper part 2. Part 1 I have already done, conducted in the year 2014. Coming to the first question. The following are the key resources needed for efficient marker assisted germplasm enhancement. So marker assisted breeding uses DNA markers associated with desirable traits to select a plant or animal for inclusion in breeding program early in its development. So now rank the key resources in the right order of requirement. So first one is established marker trait association for traits of agronomic importance. So that is the first one. This is first one. Then comes suitable characterized genetic markers and the necessary information for multiplexing. Then comes high density maps, molecular maps and densely spread markers. And then comes high throughput genotyping systems. So the answer is 3, 1, 2 and 4. 3, 1, 2 and 4 is option 1. Which of the following methods is highly amenable for automation in most of the genotyping studies? So it is SNPs. Then a patch clamp device is used to study the properties of individual membrane channels. Which one of the following is a component in the signaling pathway stimulated by receptor tyrosine kinase? So it is RAS activating protein. A mutation that inactivates the cytochrome BF complex would. So it would inhibit the movement of electrons from pigment system 2 to pigment system 1. Then how many molecules, ATP molecules are required for the conversion of one nitrogen to NH4 plus during biological nitrogen fixation? So you need 16 molecules of ATP. Answer is option 4. Which of the following bacterial gene can be used for increasing starch content in potato? So it is ADP glucose pyrophosphorylase. Answer is option 2. Then, which one of the following options describe the term transplastomics correctly? So targeting genes into chloroplast. That itself is correct. Option 1 is correct. The electrons from excited chlorophyll molecule of photosystem 2 are accepted first by. So they are accepted first by the quinone. Answer is option 4. Then which one of the following agents stimulate direct DNA uptake by protoplast? So it is polyethylene glycol. Transgenic plants expressing Barnase or Burstar genes are used for hybrid seed production. Expressing Barnase or Burstar gene are used for hybrid seed production. If you are using Barnase, it is male sterile line, and if it is Burstar, it is restoring the male sterile, restoration of the male sterile line. The presence of polyadenylation signals in the wild type Cry1AC gene from Bacillus thuringiensis prevented expression of appropriate amount of Cry1AC protein in transgenic plants. What was done to overcome this problem? So to overcome this, the Cry1AC gene sequence was modified taking help of codon degeneracy. Answer is option 3. The following are some of the genes and DNA sequences important for agrobacterium-mediated transformation of plants. Genes conferring resistance to an antibiotic under a promoter expressed in plants. tDNA border sequences. Vir genes, a reporter gene like beta-glucuronidase under CME35S promoter. Which of the above features in combination given below are present on a binary vector and minimally required for transfer of tDNA from agrobacterium to plant cell and positive selection of the transformants? So genes conferring resistance to an antibiotic under a promoter expressed in plants and tDNA border sequences. Answer is option 1, 1 and 2. Which of the following hormone ratios usually promote shoot formation from callus? So low auxin and 2 cytokinin ratio. Answer is option 2. Then which one of the following dyes can be used to test the viability of cultured plant cells? So it is fluorescein diacetate. Answer is option 1. Two Vir proteins with nuclear localization signals which help in the movement of tDNA to plant nucleus are Vir D2 and Vir E2. Answer is option 3. Which one of the following statement is not true for agrobacterium mediated plant transformation? Vir genes are essential for gene transfer. tDNA borders are essential for gene transfer. Genes for hormone and opine synthesis are essential. That is not essential for gene transfer. So answer is option 3. The breeding method for conventionally transferring cytoplasm from one genotype to another is backcross. Answer is option 3. Detasseling is a method of emasculation followed in. So it is followed in maize. So detasseling is the process of removing the tassels from the top of the corn plant to prevent it from self-pollination. So tassels are pollen producers. So detasseling is a method of emasculation. It is followed in maize. A mechanism where stamens and pistils of a hermaphrodite flower may mature at different times leading to cross-pollination is. It is dichogamy. Answer is option 2. Which of the following amino acids is an example of a compatible osmolyte in response to a range of environmental stresses? So it is proline. Answer is option 3. The principal signal molecule involved in induced systemic resistance in plants is. So it is jasmonic acid. Then international treaty in the field of the protection of the new variety of plants and rights of the breeder is. It is UPOV, International Union for the Protection of New Varieties of Plants. UPOV. Then the revised GenBank standards for plant genetic resources for food and agriculture were endorsed at the 14th regular session of the CGRFA at Rome in 2013. Gluconobacter diazotrophicus is predominantly found in endorhizosphere. Answer is option 3. A system in which there is exchange of energy but not mass is called an closed system. Answer is option 4. The second law of thermodynamics is concerned with direction of energy transfer. Option 4. An enzyme having Km values of 2.5 into 10 raised to minus 5 and 2.5 into 10 raised to minus 7 molar. This minus 5 and minus 7, they are 10 raised to minus 5 and 10 raised to minus 7. It is a typographical error. For the substrates S1 and S2 respectively is added to a solution consisting of 100 nanomoles of both S1 and S2. Which of the following statement is correct? So Km indicates the affinity of an enzyme for a given substrate. The lower Km value, higher the affinity of the enzyme for the substrate. Here S2 is having low Km value and hence higher affinity for the enzyme. So option 2, most of the active sites of the enzyme will be occupied by S2. Then next one. A digestible linear polysaccharide abundantly found in cereals having alpha-1,4 linkages in its structure is amylose. Prominent prebiotic substance is fructose oligosaccharide. Pyramidal neurons in the cerebral cortex are found in layer 3 and 5. Tay-Sachs disease is due to defect in the enzyme the GM2 ganglioside and hexosaminidase A as its substrate. Which one of the following causes stunted growth and severe fasting hypoglycemia with ketonuria? Glycogen synthetase deficiency. Then, which of the following technique is of highest resolution for detection of chromosomal alteration? CGH or comparative genome hybridization. also referred to as chromosomal microarray analysis and array CGH, is a method of genetic testing that may identify small deletions and duplications in the chromosome. Or even inversions also will be identified. Then E. coli bacteria are beneficial to humans because they produce vitamins and amino acids. Which of the following reporters can be used for magnetic resonance imaging? Transferrin receptor. Answer is option 4. Then which one of the following statement form the basis for increased circulatory lifetime for a sialylated recombinant therapeutic protein? So sialation of glycoprotein therapeutics play an important role in regulating circulatory half-life of recombinant drugs. Poorly sialylated glycoproteins with exposed galactose residues bind to asialoglycoprotein receptors expressed on liver cells, that is hepatocytes, which lead to the removal from circulation, resulting in shorter in vivo half-life of the therapeutic protein. So here they have asked which one of the following statements form basis for the increased circulatory lifetime of a sialylated recombinant protein. So it should not be recognized by asialoglycoprotein receptors present in the liver cells. If they are recognized, then they will be removed from the circulation, resulting in shorter in vivo half-life of the therapeutic protein. So option 2 is correct. Sialic acid terminated glycans are not recognized by asialoglycoprotein receptors of hepatocytes. The next one. Which one of the following play a role in changing the antigen binding site of a B cell after antigenic stimulation? So it is somatic hypermutation. Rostral is an anatomical term meaning towards the nose. Phlebitis is the inflammation of vein. Mucosal immunity is preferentially stimulated if the immunogen is administered orally. An example of lysogeny in animals could be, so viral latency or viral is the ability of a pathogenic virus to lie dormant, that is latent, within a cell, denoted as the lysogenic part of the viral life cycle. A latent viral infection is a type of persistent viral infection which is distinguished from a chronic viral infection. So answer is option 2, that is latent viral infections. Then which one of the following is the earliest site of hematopoiesis in embryo? So it is yolk sac. Answer is option 3. Which one of the following viruses contains single-stranded DNA as the genome? So it is parvovirus. Answer is option 1. Which of the following is true regarding the drugs that affects the stability of microtubules used in cancer chemotherapy? They interfere with mitosis. Answer is option 3. Therapies of lysosomal and peroxisomal disorders that have shown success in clinical trials with enzyme replacement therapy exclude. They exclude Refsum's disease. So Refsum disease is a peroxisomal disorder caused by the impaired alpha oxidation of branched chain fatty acid, resulting in buildup of phytanic acid and a derivative in the plasma and tissues. So therapies of lysosomal and peroxisomal disorders that have shown success in clinical trials with enzyme replacement therapy, they exclude Refsum's disease. Then lysosomes are thought to play an important role in which of the following processes? Class 1 MHC2 restricted antigen presentation. No, it is class 2 MHC restricted antigen presentation. Answer is option 2. Then COFAL test is used for the diagnosis of avian leukosis. Answer is option 3. Which one of the following animals has a cheek pouch in their mouth and used as an animal model for studying oral cancer? It is hamster. Then which of the following protozoan is transmitted by ingestion of ticks? Hepatozoon canis. Answer is option 3. Demyelination of the central nervous system white matter produced by canine distemper virus is an example of. It is an example of liquefactive necrosis. Which one of the following clinical condition does not have exudase? So it is granulomatous inflammation. BioSteel is a trademark for a high strength based fiber material which was made from recombinant spider silk like protein extracted from the milk of transgenic goat. Answer is option 1. Which of the following protozoan parasites replicated inside a non-nucleated human cell? It is Plasmodium. Hypophyseation refers to. It refers to injection of pituitary gland extract. The term mitotic gynogen refers to a fish that has only a female parent whose diploidy status is restored by preventing the first mitosis of oocytes. Answer is option 1. In animal cell culture, carbon dioxide incubator is used for maintaining open culture system. What is the function of carbon dioxide? It dissolves in the medium and generates carbonic acid and regulates the pH to neutrality. Answer is option 3. Then, which one of the following is a fish cell line? It is RTG2. Answer is option 3. Then, photosynthetic sulfur bacteria get hydrogen ions for carbon dioxide reduction from hydrogen sulfide. Then, white spot syndrome virus is transmitted both vertically and horizontally. Cinchonotide is a synthetic bioactive peptide originally isolated from the marine snail Conus magnus. It is used as an analgesic agent. Which of the following is a bioluminescent bacteria? It is Vibrio harveyi. Answer is option 1. The site of production of gonad inhibiting hormone in crustaceans is exorgan. In bony fishes, immunoglobulin IgM is secreted as a tetramer. Which one of the following electron acceptors used by bacteria is mainly responsible for microbial induction of marine corrosion? It is SO4 2-. Which of the following methods is used to identify the sites in a genome that are occupied in vivo by a gene regulatory protein? It is chromatin immunoprecipitation or CSIP. The class of immunoglobulin found in fish is IgM. Besides nitrogen fixation, the heterocytes of cyanobacteria also contribute to ATP production. The enzyme involved in hydrogen production from biophotolysis in green algae is FeFe hydrogenase. The compound used for the preferential removal of diatoms from microalgal cultures is germanium dioxide. Option 3. The overall reaction of microbial conversion of glucose to L-glutamic acid is this. They have given the equation. What mass of oxygen is required to produce 49 gram of glutamic acid? Molecular weight of glutamic acid is 147. So 3 by 2 O2 is 3 by 2 into 32, that is 48. For producing 147 gram of glutamic acid, you need 48 gram of oxygen. So to produce 49 grams of glutamic acid, you need 16 grams. Answer is option 1. Then which one of the following databases allow users to search marine species data sets from all of the world's ocean? It is OBIS. Option 2. Nitrogen fixation requires large amounts of energy since there is high activation energy for breaking the triple bond of the nitrogen. If so, how many molecules of ATP are required for reducing one molecule of nitrogen? That is 16. Marine environment has abundance of osmotrophs. Osmotrophs are defined as organisms which obtain nutrients and energy via passive or active transport of low molecular weight substrate across cell membrane. Option 1. Most endangered species are victims of habitat destruction. Answer is option 2. The ozone layer saves us from lethal UV. It mainly absorbs UVB. Total energy available for work at equilibrium is termed as free energy. Global warming is due to absorption of infrared radiations by carbon dioxide. In a pond ecosystem, net productivity by zooplankton is P and biomass consumed by small fishes is C. Then the ratio C by P is termed as consumption efficiency. Then two species or populations are competing for the exact same resources and one will eventually exclude the other. What is the technical term for this? It is called as competitive exclusion. Answer is option 2. Which of the following processes is most capable of slowing global warming? It is photosynthesis. Ecosystem is mainly concerned with energy flow and nutrient recycling. An organism with the ability to withstand changes to biotic and abiotic environmental factors is said to have tolerance. Option 1. An undersea volcano in the Hawaiian island chain erupts, forming a new island in the Pacific Ocean. Over the course of time, which one of these would most likely be the first species to survive on the new island? It will be lichens. They are the primary colonizers. A compound X inhibits an enzyme A competitively. A small concentration of X increases the enzyme's activity while higher concentration inhibits the activity significantly. This indicates that the enzyme is allosteric. The next one. It is observed that there is a multiple sequence alignment of homologous proteins. There is absolutely conserved glycine residues at a particular position. Crystal structure analysis of a representative protein show that the phi and psi angles of this residue occurs in the bottom right quadrant of the Ramachandran map. What is the evolutionary basis of conservation of this glycine residue? No other amino acid can occupy this position in Ramachandran map, hence mutation at this position can be structurally destabilizing. That's correct. So answer is option 1. Which of the following terms will have to be taken into consideration for developing a potentially functioning for docking simulations? Potential function for docking simulation. So docking is a computational procedure of searching for an appropriate ligand that fits both energetically and geometrically into the protein's binding site. In other words, it is the study of how two or more molecules, that is ligand and protein, fit together, that is both energetically as well as geometrically. So, which of the following terms will be taken into consideration for developing potential function for docking simulation will be hydrogen bonding, van der Waals and electrostatic interaction terms. Then, you are interested in a particular enzyme that is expressed in various human tissues. You have isolated the protein from the brain, liver and kidneys. After a lot of experimentation, you determine that the liver protein has three domains A, B, C occurring in sequential order. Domain B is the catalytic domain and the other two have regulatory function. The kidney protein has only domains A and B in that order and the brain protein has domains B and C. You then proceed to determine the primary structure of the protein using chemical methods and find that the amino acid sequence of the three domains are completely identical regardless of the source from which they were isolated. You then ask the question whether the three different proteins have all originated from the same gene by means of alternative splicing or they could be products of different genes. Having experimentally determined protein sequences and knowing the sequences of the human genome, Which one of the following bioinformatic method you will use to answer these questions above? So there are three domains for a protein, that is A, B and C. Domain B is the catalytic and the other two are regulatory, and kidney protein has only A and B domains. Brain protein has B and C. And then what you do is you determine the primary structure and find the amino acid sequence, and you find that the three domains have completely identical regardless of from where they were isolated. Then a question arises in your mind where you ask whether the three different proteins have originated from the same gene by means of alternative splicing, or they are products of different genes. So, which of the following bioinformatic method will be useful to answer the above questions? So, T-BLAST-N using protein sequence as query and human genome sequence as database. That is the answer. So, using T-BLAST-N, you can know protein sequence homology to nucleotide sequence, whether they arose from the same gene or different gene. When P, Q are length of sequences, the computational complexity of the Needleman-Wunsch algorithm is. It is OPQ. Answer is option 1. Given the following table of joint and marginal probabilities, what is the value of P B2 by A1? That I have done in the next slide. So this is it. So joint probability is the probability that two events will occur simultaneously, and marginal probability is the probability of the occurrence of a single event. Now this one is the joint probability, that is A2 B1, B by N. And this one, that is only of A1, that is the marginal probability of A1. This is the joint probability of A2 and B1. So here they have given the table. I have rewritten the table here. I have found the total also. So they have asked marginal probability of A1. So that will be, marginal probability will be 0.45 of A1, 0.45. Then they have asked for probability of B2 by A1, that is probability between B2 and A1. That is joint probability, that is 0.21. So 0.21 by probability of A1 will be 0.45. So that will give you 0.467. Answer is option 1. Then next one. In the following figure, identify the parallel and anti-parallel beta strands in the protein structure. So residues 30 to 40 is anti-parallel, that is over here. 30 to 40, they are anti-parallel beta strands, and these are the parallel beta strand, that is 50 to 60. So answer is option 1. Then E. coli ribosomal release factor gene has an in-frame stop codon in the middle of the protein coding sequence. Mutating the stop codon in this gene makes the protein non-functional. Which one of the following is an adequate explanation for this observation? So, answer is option 1. When the intracellular concentration of this protein is low, ribosomes jump over the stop codon and synthesize the full-length protein. When it is high, the protein itself facilitates release of its mRNA from the ribosome at the stop codon. Answer is option 1. Then next one. A protein has 3 domains P, Q and R, whereas another protein has 3 domains R, S, Q in that order. The preferred algorithm for these two proteins, that is in local alignment. In local alignment, only the highest scoring region is included, whereas in global alignment, all portions of the two sequences must be included in the alignment. So domain is likely only in one piece of the protein, so local alignment is preferred. Coming to the next question. A PAM 120, PAM 80, PAM 60 scoring matrices are suitable for aligning sequences with. So higher the PAM value, lower the similarity between the sequences, or lower the sequence similarity, or I can say that PAM matrix is inversely proportional to similarity between the sequences. So answer will be PAM 20 will be 40%, then PAM 80 for 50%, and PAM 60 will be 60% similarity respectively. So answer will be option 1. Then next one. Which of the following descriptors would be suitable set for quasar analysis? So descriptors are chemical characteristics of a molecule in numerical form. Example log P, which is a quantitative representation of lipophilicity of the molecule. So here they have asked descriptors suitable for QSIR analysis will be log P, molecular volume, Hammett, sigma and pi constants, molar refractivity, polar surface area. Answer is option 1. Then a closed plasmid circular plasmid of length 5000 base pair is completely relaxed in aqueous buffer. If the plasmid is put in 80% ethanol so that it transforms to A-form DNA, what will be the status of its superhelicity? So when it forms A-form DNA, number of base pair per turn becomes 11 from 10.5. So it becomes positively supercoiled. So this answer will be option 1, that is it becomes positively supercoiled. How many edges meet at the branch node in phylogenetic tree? That is 3. In pairwise alignment, an optimal alignment is one that. So an optimal alignment is either minimizes the implied number of evolutionary changes or maximizes a particular scoring function. Then which one of the following correctly specifies the order of helices according to their radius? So pi helix greater than alpha helix greater than 310 helix. Next one. In protein sequence analysis, twilight zone refers to the evolutionary distance corresponding to about 30% identity between the two proteins. Option 3. Then the double helical structure of DNA was first obtained using fiber diffraction and molecular modeling. Molecular dynamics differ from molecular mechanics by taking account of velocities of the constituent particles. An organism has 10 pair of chromosomes. If all the genes in this organism were mapped, how many linkage groups would be observed? So the total number of linkage groups in an organism equal to the number of autosomes in its haploid cell. The cell has 10 pair of chromosomes, which means that the number of autosomes in the haploid cell is 10. Hence, the total number of linkage groups will be 10. Answer is option 1. The following can be used as DNA markers: restriction fragment length polymorphism, amplified fragment length polymorphism, randomly amplified polymorphic DNA, and microsatellites. Which of the above can be used to distinguish heterozygote from a homozygote? So those which are codominant. So answer is restriction fragment length polymorphism and microsatellites. So A and D. The following events lead to changes in DNA: inversion, recombination, translocation, transition. Which of the above can lead to changes in the linkage map of an organism? That is A and C, that is inversion and translocation. The following are terms which are used to describe sequence identities: homologs, paralogs, orthologs, analogs. Which of the above can be used to describe the relationship between myoglobin gene from human and that of mouse? So orthologs are genes in different species that evolved from common ancestor gene by speciation. In general, orthologs remain the same function during the course of evolution. They are just like homologs. So answer will be option A and C, homologs and orthologs. Then to map a single linkage map in Drosophila, a 3-point test cross was carried out. The parental cross was between homozygous flies of genotype A plus C and plus B plus B plus. The double crossovers obtained after the test cross had the genotype A++ and plus CB. What is the order of the gene? So these are the parental combinations. That is A plus C and plus B plus. And these are the double crossovers A++ plus C B. So the double crossover event moves the middle allele from one sister chromatid to the other. We can see from the table that the C gene must be in the middle because the plus allele is now on the same chromosome as the A and plus alleles. See, C gene has moved from this region to this chromosome, and the C allele is on the same chromosome as the recessive plus and B alleles, and it has come along with B. Whereas, okay, so the middle gene will be the one which moves from the one sister chromatid to the other in a double crossover event. So here C will be the middle, so it will be ACB. Answer is option 3. Mouse human somatic cell hybridization led to a number of cell clones in which all mouse chromosomes are present but only certain human chromosomes are retained. The results are summarized below. The table indicates 3 such clones. Plus indicates presence of human chromosome and minus indicates those that are absent. All other human chromosomes are present. So these are the hybrid clones and these are the human chromosomes. So option, if an enzyme activity was present only in C clone, the allele encoding the enzyme is present on which chromosome? So now they have asked for enzyme activity which is present only in C but not in others. So if you take option 1, 2. So 2, it is present in both A and B. Then 3 present in but absent in C. Okay, in 3 it is absent in B but present in A and C. It should be C alone. So that also not possible. Now 6 if you take, it is present only in B but absent in C. So that is also not possible. So 7, it is absent in both A and B but present in C alone. So the enzyme activity is present only in C alone. So the answer is option 4. 7. The following pedigree shows the inheritance of an autosomal recessive trait. What is the probability that a child born to individual AB will show the trait? So this is A and this is the genotype of A, that is capital A small a and capital A small a for B. So they both will, there will be crossing and you will get about 1/4 of the progeny will show the trait. So the answer is option 2, 1/4. Then assume a population in Hardy-Weinberg equilibrium for alleles of an autosomal recessive disease locus. The frequency of the allele is 1/50. The fraction of the population representing carriers of the disease is closest to. So Q is 1/50, P is 1 minus Q, that is 49/50, or even we can write 50/50 approximately for easy calculation. So 2, they have asked the population representing carriers. So 2PQ, 2 into 49/50 into 1/50. If we assume 49 to be approximately near 250, then they will cancel out and you will get P is 1/25. 2PQ is 1/25. So answer is option 2.